Welcome to this university-level lecture on the fundamentals of biomolecules. Today, we are going to explore the fascinating world of DNA and RNA, the master architects and builders of life. We will understand not just what these molecules are, but how their physical shapes, sizes, and structural variations dictate how every single cell in your body functions and survives. Let us start by imagining DNA as a long, flexible ladder. If you twist it normally, you get a standard, comfortable staircase. But depending on the amount of water around it or its specific building blocks, this ladder can stretch, compress, or twist differently. In biological systems, DNA primarily exists in three distinct structural forms, known as aDNA, bDNA, and zDNA. Understanding this flexibility is absolutely crucial because the specific physical shape of the DNA helix directly dictates how cellular machinery reads and interacts with the genetic code. Now that we know DNA can change shape, let us look at the most common form, bDNA. This is the standard right-handed double helix found in living cells, providing perfect accessibility for enzymes that replicate our genes under normal conditions. However, when DNA is dehydrated or pairs with RNA, it compresses into aDNA. aDNA is also a right-handed helix, but it is significantly shorter and wider. This temporary wider shape is absolutely crucial during the process of making proteins in the cell. Moving beyond the standard right-handed forms, we encounter zDNA, which is a radical departure from the others. zDNA is a left-handed helix with a distinct stretched zigzag backbone. You might wonder why the cell needs such a drastically different and unusual shape. It is believed that zDNA acts like a physical switch, playing a vital role in regulating gene expression by turning certain genes on or off depending on the immediate metabolic needs of the cell. To truly understand the physical differences between these DNA types, we can calculate their exact lengths. The total length of the DNA helix depends on the number of base pairs and the vertical distance each pair adds, known as the axial rise. In this formula, L represents the total length. N stands for the number of base pairs, and H is the axial rise. For standard bDNA, H is approximately 0.34 nanometers. If the DNA compresses into aDNA, this rise decreases, meaning the overall length will shrink significantly, even if the number of base pairs remains exactly the same. Before we move on, let us see this formula in action with a concrete example. Imagine a scientist studying a specific gene consisting of exactly 2,000 base pairs in the standard bDNA conformation. We want to find its total physical length. We substitute 2,000 for N and 0.34 for H, which is the standard rise for bDNA. Multiplying these together gives us exactly 680 nanometers. This means if we stretched this specific gene perfectly straight, it would measure 680 nanometers from end to end. Now, let us see what happens when this exact same DNA segment is exposed to severe dehydration, causing it to transition into aDNA. The number of base pairs remains 2,000, but the axial rise drops to 0.26 nanometers. Multiplying these gives a new length of 520 nanometers. To find the exact amount of physical compression, we subtract this new length from our original bDNA length. The result is 160 nanometers. This significant reduction perfectly illustrates how environmental changes can drastically alter the physical dimensions of a biomolecule. Let us visualize these structural variations we just calculated. On screen, you can see the three distinct forms of DNA side by side. In the center is the standard bDNA, which is the most common form under high hydration. Notice how dehydration forces it to compress into the wider, shorter aDNA on the left, exactly as our math showed. On the right, you see the radical zDNA, which twists in the opposite, left-handed direction, with a stretched, zigzag backbone. This diagram beautifully summarizes how DNA dynamically shifts its shape depending on its immediate environment. Having explored the architect of the cell, DNA, we now turn to the messengers and builders, RNA. If DNA holds the master blueprints locked safely in the nucleus, RNA represents the photocopies, the workers, and the tools that actually build the proteins. Ribonucleic acid, or RNA, is typically a single-stranded molecule. Unlike DNA, it contains the sugar ribose and uses the nitrogenous base uracil instead of thymine. Without RNA, the genetic information stored in your DNA would be completely useless to the cell. To build a protein, the cell relies on three primary types of RNA, each with a distinct role. First, messenger RNA, or mRNA, acts as the expendable middleman, carrying the genetic code from the nucleus to the cytoplasm. Next, transfer RNA, or tRNA, acts as an adapter. It is a small cloverleaf-shaped molecule that brings specific amino acids to the construction site. Finally, ribosomal RNA, or rRNA, forms the actual factory machinery of the ribosome itself, physically linking the amino acids together. They are the unsung heroes of the cell. Because RNA is typically single-stranded, we can easily estimate its total molecular weight if we know how many building blocks it contains. This brings us to a very simple but powerful formula. W represents the total estimated molecular weight, measured in Daltons. N stands for the total number of nucleotides present in the RNA sequence. We multiply N by 330, which is the average molecular weight of a single RNA nucleotide. If the length of the RNA transcript increases, its total molecular weight will increase proportionally, allowing us to quantify its size. Let us apply this formula to a specific messenger RNA molecule, freshly transcribed from a gene. Researchers find that this mRNA is exactly 1,220 nucleotides long. We want to determine its estimated molecular weight. We replace N with 1,200 and multiply it by our constant, 330. The resulting product is 396,000 Daltons, often written as 396 kilodaltons. Knowing this precise weight allows scientists to verify if they have successfully isolated the correct mRNA in the laboratory. Now that we know the size of the messenger RNA, we can predict the size of the final protein it will produce. During translation, the ribosome reads the mRNA sequence in groups of 3 nucleotides, called codons. Each codon codes for exactly 1 amino acid. Therefore, the number of amino acids, A, equals the total number of nucleotides, N, divided by 3. This simple division formula perfectly mirrors how the ribosome physically steps along the mRNA strand, reading the genetic message triplet by triplet to build the final protein structure. Let us look at the exact same mRNA molecule from our previous example, which is 1,200 nucleotides long. how many amino acids will be in the final assembled protein? We substitute 1200 for N and divide by 3. The math is straightforward, giving us exactly 400 amino acids. This beautifully demonstrates the direct, predictable mathematical relationship between the length of the messenger RNA and the size of the functional protein it creates. You can now predict the physical scale of a protein just by looking at its genetic blueprint. Let us bring all these RNA types together in this diagram of the translation process. The long horizontal strand is the messenger RNA, carrying the instructions. The large structure clamped around it is the ribosome, made of ribosomal RNA, which acts as the factory floor. Notice the clover-leaf-shaped transfer RNA molecules arriving. They are reading the mRNA codons and delivering the correct amino acids. As the ribosome moves along the mRNA, it links these amino acids into a growing polypeptide chain. This is the dynamic workforce of the cell in action. We have covered a lot of ground today, so let us summarize the most important ideas. First, DNA is not a rigid molecule. It dynamically shifts between A, B, and Z forms, and this physical shape directly controls how genetic information is accessed. Second, we saw how the 3 types of RNA, messenger, transfer, and ribosomal, work together to bridge the gap between static DNA blueprints and living proteins. Finally, we learned how simple math allows us to predict the physical dimensions of these vital biomolecules. You now have a strong foundation in the fundamentals of DNA and RNA.